Dear student, our lecture today is about mycoplasma. Here are some key definitions that will be mentioned during the lecture. Learning objectives of the lecture: overview about mycoplasma, general features of mycoplasmas, classification, Mycoplasma pneumoniae, genital mycoplasmas. Now overview about mycoplasma. Mycoplasma are small prokaryotic organisms with no peptidoglycan in their cell wall, enclosed in a single plasma membrane. Peptidoglycan, as you know from previous lecture, is a polymer consisting of a sugar, amino acids that form a mesh-like layer outside plasma membrane of most bacteria. Many mycoplasma species include several common cells commonly found in the mouth and genitourinary tract of humans. Remember, do not confuse Mycoplasma species with Mycobacterium species. The general features of mycoplasmas: plastic and pleomorphic organisms due to the lacking of cell wall. Pleomorphic organisms mean that have ability to alter their morphology, biological functions, or reproductive modes in response to environmental conditions. Mycoplasmas cannot classify for this reason as either cocci or rods. The smallest of non-free living, self-replicating prokaryotic cells. Self-replicating prokaryotic cells. Cells that can replicate although it contains the minimum set of organelles essentials for growth and replication, such as plasma membrane, ribosomes, and genome consisting of double-stranded circular DNA. As we mentioned, that mycoplasmas have very small double-stranded DNA genome. Also, mycoplasmas require a variety of small organic molecules for growth. Mycoplasma contains steroids in their cell membrane. Sterols are subgroups of steroid with hydroxyl group at the 3 position of a ring. They are amphipathic lipids. The overall molecule is flat and the hydroxyl group on a ring is polar. Mycoplasma, rarely cultured in the lab, produce minute colony on specialized agar after several days of incubation. The central portion of the colony penetrates the agar, whereas the periphery spread over the adjacent surface. It resembles fried egg appearance. Classification of mycoplasma. Mycoplasma is classified to Mycoplasma pneumoniae, which cause lower respiratory tract infection, atypical pneumonia, Mycoplasma hominis, uroloticum, and genitalium. All these three cause genital mycoplasmas, often associated with sexually transmitted infections. Also, infections occur during childbirth. Pathogenesis of Mycoplasma pneumoniae. Mycoplasma pneumoniae possesses a membrane-associated protein P1, which binds sialic acid-rich glycolipids found on certain host cell membranes, ciliated bronchial epithelium cell in that case. Then it grows closely attached to the host cell luminal surface and inhibits cellular reaction. The pouches of affected mucosa desquamate, that means it becomes scaly, and inflammatory response develops in bronchial and adjacent tissues involving lymphocytes and other mononuclear cells. Note that Mycoplasma pneumoniae produces exotoxins that result in extensive vacuolization and death of host tissue. Vacuolization means formation of vacuoles or vacuole-like structures within or adjacent to cell. Now, the immune response against Mycoplasma pneumoniae elicit both local and systemic immune responses. Serum antibody to outer membrane glycolipid and to P1 adhesion can be demonstrated. IgM, called agglutinin, is produced by approximately 60% of infected patients. Now, laboratory diagnosis. Serological tests mostly used for diagnosis of atypical pneumonia. Genital mycoplasmas, Mycoplasma hominis and Ureaplasma ureticum. These are common inhabitants of genitourinary tract, particularly in active adults. In some population, colonization rate is 50%. Both can be cultured. They grow more rapidly than Mycoplasma pneumoniae. Hominis and Ureaticum can be distinguished by their carbon utilization pattern. Hominis degrade arginine while Ureaticum hydrolyzes urea. Mycoplasma hominis isolates are resistant to erythromycin in contrast to other mycoplasmas. Ureaticum is a common cause of urethritis when neither gonococcus nor chlamydia can be demonstrated, particularly in men. Urethritis is inflammation of urethra. Urethra, the tube that carries urine from bladder to outside the body. While in women, ureaticum has been isolated from the endometrium of patients with endometriosis and from vaginal secretion of women who undergo premature labor or deliver low-birth weight babies. Endometriosis is the inflammation of lining of uterus. Ureaticum also isolated from the infant lower respiratory tract and CNS, both with or and without evidence of inflammatory response. Mycoplasma genitalium has been recognized as a sexually transmitted pathogen, resulting in a series of syndromes similar to those caused by Neisseria gonorrhea and Chlamydia trichomonas. Once upon a time, Neisseria gonorrhea was a major cause of urethritis. The cases of urethritis that were not caused by Neisseria gonorrhea were referred to as non-gonococcal urethritis (NGU). Today, the major cause of NGU is Chlamydia trachomatis, but other organisms now appear, which is Mycoplasma genitalium and Ureaplasma urethicum, also cause non-gonococcal urethritis. The diagnosis is done by using PCR amplification is recommended for specific diagnosis in that case. Extraction of the DNA and amplification and detection by real-time PCR or PCR, conventional PCR. Now here are some study questions. I hope you read these questions and try MCQ and try to solve it, and then we might discuss between us in Telegram, or you can discuss with your colleagues and try to solve these MCQ questions. Thank you.